Hello students, this is Mrs. Sonal Patil. In our last class, we have studied about outbreeding devices, pollen-pistil interaction, artificial hybridization, and we ended up with double fertilization. Today, we will continue the remaining part of the chapter. We will start first with the significance of double fertilization. As I told you, double fertilization in angiosperm results in the formation of the zygote and primary endosperm nucleus. The zygote develops the embryo and the primary endosperm nucleus develops into endosperm. So zygote develops into embryo and primary endosperm nucleus develops into endosperm. Now this endosperm is a nutritive tissue which nourishes the developing embryo. So remember, endosperm is a nutritive tissue which is nourishing the developing embryo, which is formed by the development of zygote. The endosperm in angiosperm is triploid and it is a post-fertilization structure, but in gymnosperms it is haploid and a pre-fertilization structure where the embryo is formed later. In gymnosperms, when there is no fertilization and embryo formation, the nutritive tissue is simply wasted, while in angiosperms this wastage is avoided because of its post-fertilization endosperm formation. Fertilization gives necessary stimulus to the growth of embryo, leading to fruit formation. The zygote formed by the fusion of male gamete and egg cell helps to maintain the constant chromosome number in the life cycle of the plant. It also helps in the recombination of genes and development of new characters in the offspring, which may lead to origin of new species. So this is the significance of double fertilization or the importance of double fertilization. Again remember, double fertilization is an important concept to be asked in the exam. So very important from exam point of view. Coming to the next part, we have already discussed earlier the pre-fertilization structure and events. Now we have to study post-fertilization structure and events, which include endosperm and embryo. Endosperm, as I told you, endosperm is a nutritive tissue which is nourishing the developing embryo. Okay, so how it is formed? The primary endosperm nucleus undergoes a series of division and ultimately forms endosperm, which is highly nutritive tissue which provides nourishment to the developing embryo. Okay, so as I told you, here during double fertilization what is happening is that the two male gametes are released. Okay, one male gamete fuses with the egg cell forming zygote, another male gamete fuses with the polar nuclei and forms primary endosperm nucleus, which later develops into endosperm. Now these endosperms are of different types. On the basis of development, three types of endosperms have been recognized. Firstly, nuclear endosperm. In this type of development, the primary endosperm nucleus undergoes successive nuclear division to give rise to free nuclei. The first few cell divisions are not accompanied by cell wall formation. Nuclei produced remain free in the cytoplasm of the embryo sac. So here what is happening is that the primary endosperm nucleus undergoes successive nuclear division to give rise to free nuclei. A few thousands of nuclei which are produced are pushed towards the periphery of the embryo sac. The central part of the embryo sac thus has a large vacuole and forms the nuclear endosperm. The example, very good example over here is Cocos nucifera, that is coconut. The watery liquid endosperm which fills the large embryo sac contains numerous free nuclei. So, the coconut water from tender coconut is called free nuclear endosperm. Later, cell wall formation occurs around the nuclei at the periphery to form coconut meat. The quantity of the cellular endosperm increases by the division of cells to form the coconut kernel. In mature coconuts, the liquid endosperm becomes milky and it does not contain free nuclei or cell. So, this is the nuclear endosperm. The second one is cellular endosperm. Wall formation occurs with first division of primary endosperm nucleus. After first transverse division, the subsequent division are irregular. You can see over here the nuclear divisions here are nothing but they are followed by transverse division over here, and then the endosperm tissue cell do not show a regular arrangement. It is not showing regular arrangement in the cellular endosperm. So, here is the primary endosperm nucleus undergoes transverse division and then the divisions are irregular, and finally the endosperm tissue cell do not show the it is not showing regular arrangement. This is called as cellular endosperm. The third type is halobial endosperm. This type is intermediate between the nuclear and cellular type. The first division of primary endosperm nucleus is accompanied by formation of transverse wall. This divides the embryo sac unequally into a small chalazal chamber and a larger micropylar chamber. You can see here transverse division has occurred and it has resulted in the formation of micropylar cell and a chalazal chamber or the chalazal cell. This is followed by free nuclear division in each chamber, and then cell division or the cell wall formation make the endosperm cellular. It is making it cellular. So, it is here what is happening is that firstly the division is transverse, resulting into the formation of small chalazal chamber and a larger micropylar chamber, and later it is followed by free nuclear division in each chamber, and then the cell wall formation make the endosperm cellular. This is halobial endosperm. Next, coming to the next post-fertilization structure is the embryo. Embryo, as I told you, it is formed by the fusion of male gamete and the egg cell. Now, after fertilization, the zygote takes a period of rest, then grows and gives rise to the embryo. The process of development of mature embryo from diploid zygote is called embryogenesis. A very, very important question again for 1 mark. So, the process of development of mature embryo from diploid zygote is called embryogenesis. Most zygotes divide only after certain amount of endosperm is formed. This is an adaptation to provide assured nutrition to the developing embryo. Though the seeds differ greatly, the early stages of embryo development are similar in both monocots and dicots. A typical dicotyledonous embryo consists of an embryonal axis and 2 cotyledons. The portion of embryonal axis above the level of cotyledon is the epicotyle, which terminates with the plumule or stem tip. The cylindrical portion below the level of cotyledons is hypocotyle that terminates at its lower end in the radical or root tip. The root tip is covered with a root cap. So, this is a typical dicot embryo. Let us see how this is formed. So, development of dicot embryo. Now, how the embryo is developed, we will see. In earlier stages of embryo development, there are no fundamental differences between embryos of dicot and monocots, but marked differences are noted in later development and mature structure of their embryo sac. Now, here in dicots, zygote divides into two unequal cells, larger suspensor cell towards micropyle and a smaller embryonal cell towards antipodal region. The suspensor cell undergoes transverse division forming 6 to 10 celled long suspensor. The first cell of the suspensor is large and called haustorium or vesicular cell. The last cell of suspensor is known as hypophysis. It forms a radical tip. Embryonal cell divides twice vertically and once transversely to produce a two-tiered 8-celled embryo. The epibasal tire forms two cotyledons and a plumule, while the hypobasal tire produces only hypocotyle and most of the radicle. For this, the octant embryo undergoes periclinal divisions producing procambium and ground meristem. It is initially globular, but with the growth of cotyledons it becomes heart-shaped and then assumes a typical shape. Example is Capsella bursa pastoris, and I already explained to you all about the dicot embryo having two cotyledons, plumule, this is hypocotyle, radicle and the root cap. So, it is having typically, this is the structure of the dicot embryo. Diagram again is very important from exam point of view. You have to practice the diagram of the dicot embryo. Here the development is been shown, you can see over here. Okay, so zygote divides here, suspensor cell has been shown, and you can just see over here how it is changing to globular stage and then into the heart-shaped embryo, and finally it forms the mature embryo. So, as I already explained to you all, the earlier stages of embryo development, there are no fundamental differences between embryos of dicot and monocot, but marked differences are noted in the later development, as I told you. The first division of zygote is transverse, as already explained to you, it is transverse and forms what it is forming. It is transverse and produces two-celled pro-embryo. It is forming two-celled pro-embryo which will again divide and then finally forms the globular stage. This is the suspensor, has been shown, suspensor, and then globular stage, heart-shaped embryo, and then finally the mature embryo. This is the development of the dicot embryo. Coming to the next, the development of monocot embryo. The early development, as I discussed, of dicot and monocot embryos are similar up to octant stage. Later on, differentiation starts. Suspensor is single celled over here. The zygote divides transversely, producing a vesicular suspensor cell towards micropylar end and embryo cell towards the chalazal end. The embryo cell divides transversely again into a terminal and a middle cell. The terminal cell divides vertically and transversely into globular embryo. It forms a massive cotyledon and a plumule. Growth of cotyledon pushes the plumule to one side. Remains of second cotyledon occur in some grasses. It is called epiblast. The single cotyledon of monocot is called scutellum. It is called as scutellum. It is shield shaped and lateral in position but appears terminal. The middle cell gives rise to hypocotyle and radicle. It may add a few cells to the suspensor. Both radicle and plumule develop covering sheets called coleorhiza and coleoptile respectively. So here you can see coleorhiza and then coleoptile. So coleorhiza and the coleoptile are the coverings which are covering the radicle and plumule respectively. And these are the assignment questions. Describe the structure of dicot embryo with a neat label diagram. Secondly, describe the structure of monocot embryo with a neat label diagram. So these two diagrams along with the explanation are very important. Both dicot embryo you have to practice and the monocot embryo diagram with the description is to be practiced for 5 marks. Thirdly, what is endosperm? How does endosperm in angiosperm become triploid? Describe 3 types of endosperm on the basis of development. So these were the questions from this part of the chapter. So here today we have covered about the post-fertilization structure and events, where we studied endosperm, then we studied types of endosperm, that is nuclear endosperm, cellular endosperm and halobale endosperm, and later we studied embryo, how it is formed, and then we have studied about the development of monocot embryo and the development of dicot embryo. And for today we will stop up till the development of monocot embryo. In the next class we will start, or we will complete the chapter. We are going to start with the seeds, fruits, and finally we will end with apomixis and polyembryony. That will end the chapter. For today we have completed up till this. So thank you, and the later part of the chapter we will continue in the next class.